Hello friend, before watching my lectures, I wish subscribe my YouTube channel and comment in the comment section, hit the like button. If you have suggestion, question, you can ask me. So let's come to the topic. Our topic is neuron anatomy and function. The first lecture of neurophysiology. So let's come to the topic. There is a neuron. Neuron has its some important branch like dendrite, cell body, axon. This extension is called dendrite. The tech originate from the cell body. So the number of dendrite in this neuron is different. The middle portion is called cell body. We can easily say another name to the cell body, soma. The portion between this axon and cell body, there is axon helix. Axon helix. So this portion is called axon. The end portion of axon it is called axon terminal. So the function of neuron is to take information and transmit into the cell body after processing, transmit or transport into the axon terminal bulb. So how dendrite take information? So suppose there is a neuron, and this neuron release some neurotransmitter. So how this neurotransmitter stimulate the cell membrane of this dendrite? So if this neuron release neurotransmitter, it will stimulate or it will produce action potential within this dendrite. So we take this portion and zoom it here. So let's suppose this is the cell membrane of this dendrite. How produce its action potential? So there is some channels. The first one is a ligand gated ion channel. It will produce so quick response. The ligand gated ion channel will produce quick response. So suppose there is a neuron, and neuron release its neurotransmitter. This neurotransmitter will act in the receptor site of this ion channels, and this channel will open and the positive charge will increase within the cell membrane, of this dendritic portion. So if positive charge will, the cell will go into depolarizing stage. Because of that, we can say to this state excitatory post-synaptic potential. Excitatory post-synaptic potential. There is another channel. The function of this channel is also if the neurotransmitter released from another neuron, neurotransmitter will bind into the receptor side of this channel. This channel will also open. If this channel open, the negative charge will increase within this cell. So if the negative charge will increase, so this cell will go into the hyperpolarization stage, or the cell membrane will go into the hyperpolarization state. So we will say in this state also inhibitory post-synaptic potential. The combination of inhibitory post-synaptic potential in excitatory post-synaptic potential is called graded potential. So graded potential may be inhibitory postsynaptic potential or excitatory postsynaptic potential. These two channels are ligand-gated ion channel. These inhibitory or excitatory or ligand-gated ion channel. There is another receptor by the name of GPCR or G protein coupled receptor. This receptor is so slow. It will transmit the impulse so slowly. So how it produce action potential? When this neuron's axon release neurotransmitter, it will bind within receptor site of this GPCR or G protein coupled receptor. It will activate secondary messenger. Secondary messenger will activate protein kinase. Protein kinase also activate or stimulate this another channel. This channel may be inhibitory postsynaptic potential or excitatory postsynaptic potential. If another neuron stimulate the cell body or the soma, so which action will happen within the cell membrane of cell body? So let's suppose this is a neuron in its axon branch of this neuron. It will release neurotransmitter, and neurotransmitter will bind to the cell membrane of cell body. So there will also produce some action potential, like how produced within the cell membrane of dendrite. Same action potential will also happen there. Here is also graded potential will produce. With graded potential, there is also produce protein synthesis. If action potential produce here, it will stimulate the DNA to copy messenger RNA. In messenger RNA will make some protein vesicles within the rough endoplasmic reticulum, or the messenger RNA with ribosome will make some protein vesicle within rough endoplasmic reticulum. We can also say to these vesicle nasal body. So then nasal body will enter into the Golgi apparatus with more processing. After more processing, it will release this vesicle. The component of this vesicle may be enzyme, neurotransmitter, and membrane protein. This vesicle may contain enzyme, neurotransmitter, and membrane protein for more function of neuron. Within the axon, there is also some changes will occur. So let's see this doll by the name of kinesin. This doll will transport some mitochondria and some vesicle. Transport these mitochondria and vesicle towards the axon terminal bulb. So this protein name is called kinesin. Kinesin will transport membrane and vesicle towards the axon terminal bulb. So in this state, we will say anterograde axonal transport. If kinesin transport the mitochondria and vesicle toward the axon terminal, we will say anterograde axonal transport. There is another doll. This red line may be microtubule. So there is another doll. It will transport from axonal terminal towards the cell body. we can easily say retrograde. So this doll name is dynein. Dynein. This will also transport some mitochondria, some vesicle, some growth factor, some nerve growth factor. So it may also transport some poliovirus, rabies, herpes simplex virus, and varicella zoster virus within the axon for the cell body. If action potential reach to the axon hillock, it will go towards the axon and open some sodium channel. Sodium channel will enter into the axon, and the cell will go into the depolarizing stage. In exact time, there is also open potassium channel, and the potassium will get out from the cell, and the cell will go into the repolarizing stage. So there is a channel by the name of voltage gated calcium channel. So this positive ion will stimulate this channel. Say that if action potential reach to the axon terminal, it will open a voltage gated calcium channel. More calcium will enter to the axon terminal bulb, and calcium function is to attach the vesicle into the cell membrane. How calcium act? So let's suppose there is some protein by the name of V-Snares protein that attached to the vesicles. There is another protein that's by the name of T-Snares protein that attached to the cell membrane. These two proteins will bind with each other by calcium. So calcium attach these two proteins, and vesicle will easily attach to the cell membrane. Then vesicle will release its content or neurotransmitter within the synaptic cleft, and neurotransmitter will act into the postsynaptic neuron. If neurotransmitter act on postsynaptic neuron, it may will be inhibitory or excitatory, or it will may be excitatory postsynaptic potential or maybe inhibitory postsynaptic potential. After action of neurotransmitter, it will degrade, or neurotransmitter will terminate by two mechanisms: by reuptake and by degradation. Degradation may occur by some enzyme, and reuptake may occur by some reuptake protein channel that is located into the axon terminal bulb. It will bring the neurotransmitter into the axon terminal bulb, or these neurotransmitter will bind to the vesicle to make further vesicle.